Welcome to our Overcoming Product Shelf Life Challenges webinar hosted by New Tech Group. My name is Orna, and I will be your host today. Just a few words about New Tech Group before we move on to the webinar. New Tech works closely with quality control and research and development laboratory professionals within varied industries such as food, feed, pharma, and agriculture. Our solutions help automate and alleviate many of the common repetitive, tedious practices commonly used in every step of the quality control lab, from sample prep through agar pouring and colony counting. Other solutions can be used for analysis of water activity, color, shape, and size, as you can see in the image on the screen. Many of our customers have shared with us challenges with shelf life issues, and through our consulting services, we were able to assist in overcoming these issues and helping them achieve their goals. In our webinar today, we're going to discuss a few of these case studies. I'd like to introduce our senior application scientist, Dr. Brady Carter. Hi, Brady.
Hello.
We hear you. Dr. Carter specializes in water activity and microbial safety. He earned his PhD and master's degree in food engineering and crop science from Washington State University. He has 20 years of experience in research and development and has authored numerous white papers, has vast experience in instructing and training in these areas, both locally and internationally. He's participated in hundreds of extension presentations and has given talks at numerous scientific conferences. Just before we move into the presentation itself, just make sure that you're on mute, that your camera's off. You're going to be seeing Brady and myself throughout the presentation. After the presentation, we plan on having some time for Q&A. If you have questions during the presentation, don't hesitate to post them in the chat area, and Brady will do his best to answer when he's done. At the end of the presentation, we're going to post a short link, and we ask you to take a few minutes to complete it. Take it from here, Brady.
Okay, well, thank you so much, and thank you everybody for being here today. I hope this will be a beneficial presentation and will be something that's useful to you, and you'll be able to take some good information. So our topic is dealing with overcoming shelf life challenges for products. And if you deal with manufactured products, then shelf life, of course, is one of the things that you deal with as well. Products are made, and when products are made and manufactured, they're put into the most chaotic state they could possibly be, and then they slowly over time will relax back into their most stable condition. And as a consequence, during those while those changes are happening, properties of the product change, and which will result in their losing their desirable properties, which then ends shelf life. So what are some of the challenges that are associated with shelf life then? It's something that most people have to deal with. But in terms of determining what the shelf life of a product is, one of the first things that has to be done is to actually do some testing and figure out how long will the product last. And sometimes this step is the one, it's the most important and initial step, but it's one that's often not done because of challenges associated with trying to do shelf life testing. It takes a lot of resources, it takes a lot of time. And so what often happens is that with new products, folks will rely on their knowledge of the products or similar types of products, and will set the shelf life to a similar value. Or if there's other products that are very similar already in the marketplace, they'll just match whatever that shelf life is. So in a lot of cases, these best by dates that are reported aren't necessarily based on actual testing. They're more based on a best guess of what a conservative guess of how long the product will last, when in fact it may last longer, and be able to figure that out if they actually did shelf life testing. So what often happens then because of this is that what we end up seeing is a product that's failed when it wasn't supposed to fail after production. And when that happens, it of course sparks a lot of concern and a lot of action, if you will, because there's there's product in the field that has failed, and you got to figure out why did it fail? What was going on? What were the problems? And why do these failures happen in a lot of cases, and even when specifications are met? What steps can you take to make sure that they stay safe so that doesn't happen again? How do you determine the shelf life of your product? What ends your shelf life? What mode of failure is resulting in that product no longer being acceptable? What tests should you run when you're looking at shelf life, and can you speed up the testing? Products that have 2 years of shelf life, doing a real-time test on those is obviously very laborious and takes up space. You got to watch and wait for a year, 2 years. Can't release the product. So how can you overcome that? And so we want to see how we can possibly help New Tech Group. How can we provide some assistance with these challenges associated with shelf life? So I want to split up our discussion in basically 2 parts. Part 1, I want to talk about some actual case studies of where a product has failed unexpectedly, and how we were able to help these groups figure out why it failed, and also to put in some safety steps to make sure that it doesn't happen again. And so we'll do several different case studies showing examples of where this has happened and how we went about trying to figure it out and solve it. And then part 2 will be some actual instruction and information on how to conduct shelf life testing in a more reasonable and feasible way, and maybe provide some insights into ways that you can make that shelf life testing process more efficient. So the first example I want to talk about is a situation where pet food went moldy unexpectedly. So this is a situation where this company has made pet food for an extended amount of time, and they've never had any problems with their product. They always made it to a 10% moisture content specification. They send it out. It's a great product, a great reputation. So they're real happy with the product. So no problems for over 20 years making this product to a 10% moisture content specification. Then all of a sudden, they start getting customer complaints, and the complaints are about moldy product. And so they have to recall this product because obviously consumers, when they open it, don't want to see mold on their pet food. And even beyond that, there's a safety factor, because if there's mold, there's a good chance that there's also mycotoxin production, and mycotoxins can be very dangerous to dogs or other pets. And so they had to immediately do a product recall on this material that was going moldy. And of course, that comes with all kinds of challenges. So how did this happen? The moisture content specification didn't change. They were still making it to 10% moisture content, no problems, still making it to 10%. All of a sudden, moldy product and recall. So how does this happen? So let's first talk about what the consequences are. In this particular case, the pain comes very quickly when these things happen. The cost of the product recall up towards $10 million. An additional $5 million in lost revenue due to sales that were canceled because word had traveled around that the product was moldy. These reputation problems had to be addressed immediately. So they had to hire a PR group to handle all responses and what they were going to say in the media to make sure that that this was done in such a way to try and save their reputation. And there for a while, because that 10% moisture content specification wasn't working, they were meeting it and having bad product. They were kind of running blind, and not even sure if they could release product. Had to overcompensate and make that moisture spec very low to try and minimize the chance of having problems, and in so doing would have had other quality issues come up because they're over drying the product. So what's going on here? Well, the first thing to understand, and the first thing that we discussed when we did work and help them, was that moisture content that has nothing to do with microbial growth. Microbial growth is controlled completely by water activity. Microorganisms don't care how much water is present, they care whether they can access it. And so we can actually identify growth limits, water activity growth limits for these different organisms, where if the water activity gets below that value, then those organisms aren't able to grow. So we see here, and this would be specific to each type of species, but we can kind of group them in terms of these limits. So at 9.1 gram negative bacteria stop, 8.6 gram positive bacteria, 8H yeast, 0.80 mycotoxin production stops. Molds, their practical limit is 0.7, and then at 0.6 is the absolute limit for all growth. And this distinction between mycotoxins and mold is really critical, and was critical for this group, because come to find out that even though they weren't measuring water activity, when we checked water activity afterwards, it was found that the water activity was above 0.7, but not up to 0.8, which means that even though this product was going moldy, there wasn't any mycotoxin production. That was still too low for mycotoxin production. And so that was actually good news for them, that they were able to show that there wasn't going to be a mycotoxin problem. So let's get more in and look at why water activity is what controls microbial growth and not moisture content. Microorganisms are basically membranes surrounding sacs of solute that are out of a certain energy of water. Water activity is the energy of water. If you've participated in any of our other webinars on water activity or the theory of water activity, that's one of the most critical things to understand, is that water activity is the energy of water. It gets a lot of terms used with it, like free water and bound water, available water, but what it actually is, rooted in thermodynamics, is the energy of water. And the reason why it controls microbial growth is that these microorganisms, inside the cell, they have a certain water activity or energy of water. And they have to get water into their cell to maintain their turgor pressure, which then determines whether or not they have normal metabolic activity. And so when they encounter an environment where the water activity is lower than their interior water activity, in other words, the energy of the water outside their cell is lower than the inside their cell, water moves from high energy to low energy, which means water starts to move out of these cells, and consequently, there's a loss in turgor pressure. And so they shut down. They can't go through their metabolic activities the way that they normally would. And that's called osmotic stress. So they're being stressed because there's water leaving their cell because of this control of water moving from high energy to low energy. Now, once that happens, the cells or each of these organisms have the ability to respond. They have capabilities to try and concentrate solutes in their interior to lower their interior water activity. And that's through the concentration of polyols, sugars, kind of similar to things that are often used in products to lower water activity. But each organism has its own unique ability to be able to do this, to lower the water activity. And that's why each of them have a different water activity cutoff. It all depends on how well can they lower their internal water activity. If they're able to get lower than their environment, then water starts to move in and they start to grow. If they can't, then they're forced to go into dormancy. And these organisms can each have their kind of own unique abilities to be dormant. Some are very resilient. They form spores, Salmonella being one of the most famous, and can last for a very long time at low water activity. So water activity isn't a kill step, but it controls growth through the control of access to water by these microorganisms due to energy differences. So with that in mind, the main reason that can be seen that the failure happened in this case was because water activity wasn't a primary measurement they were making. They were trying to release product off of moisture content. Now, over time, in a lot of different situations, folks have been able to figure out what moisture content is associated with the safe water activity. And so once that's through trial and error has been figured out, then if you process product to that moisture content, then typically you're going to be okay because it's corresponds with the safe water activity. As you can see in this chart, at the lower at in this case 9% moisture, it's associated with water activity less than 0.7, and so that's product safe. Where the breakdown happened was that in this case, there was a switch in the method being used to measure moisture. And because moisture doesn't have actual standards, it's actually quite difficult to make sure that you're going to get the same results. And so in this case, through how the settings were done on the instrument, they were getting values that matched what they thought their moisture content should be at that 10%. But in reality, the moisture was higher than that, even though the instrument was telling them that it was within specification. In reality, it was higher. And when it at that higher moisture content, of course, that was now associated with a water activity well above 0.7. So that product is now going to mold. So if they had been measuring water activity as the primary release specification to assure safety and not moisture content, the problem would have never happened because water activity you can check with standards and it's what's actually controlling the growth. So they would have known, even though this was saying the same moisture level, they would have known had they been measuring water activity that all of a sudden water activity was now above 0.7 and that product was going to have problems. It was going to mold. And so then they could have made sure that that product was released and would have saved millions of dollars in lost revenue and lost costs. So the solution is to track water activity. So in this case, what was critical was for them to start measuring water activity on all of their production and releasing based on water activity being less than 0.7, set it at 0.65. And as long as it meets that specification, their product should not have any problems with mold. And regardless of what moisture content they have that's associated with that water activity, which then actually gave them an opportunity where if they reformulate and make sure that they maintain that same water activity through reformulation, they can actually increase the amount of moisture held at that safe water activity. And so now instead of being at 10%, maybe they could get up to 14%. And selling a product on a weight basis, the more water you can keep in there being your cheapest ingredient, the more money you can make. So our recommendations were to adjust their specification to release only product whose water activity was less than 0.68, and the moisture content becomes a standard of identity only. And that's really what its purpose should be. A lot of sales and a lot of transactions that happen in food are based on moisture. So it really is still something you have to measure and report, but there's no reason, and you would never want to release a product based on a moisture content as opposed to a water activity. Now, moving on to our next example. Sorry about that, the wonders of working from home. The next example is a baked snack that's was, again, a nice new product that was being made. They were making the product without necessarily seeing a lot of problems, but then all of a sudden they started getting complaints from customers that the product was showing up rancid. And so that typical rancid smell, and of course, if that happens when they open up these bulk bags to use this as an ingredient, and that smell happens, they of course are going to reject it and not put it into whatever it is that they're making. And so immediately there were complaints coming back that this was going rancid. And so the immediate thought and intuitive thought, if you will, was for this company to lower the water activity specification or sorry, lower the moisture content specification. And again, in this example, they were not releasing off of water activity or measuring water activity on the product. So instead, they lowered the moisture content specification, thinking that if they lower it, it should be more stable and they won't have problems. But in fact, they started to see even more complaints when they lowered that moisture content specification. So again, the question is, why is that the case? Here again, there was a lot of pain associated with these problems. This was an ingredient that was being sold to lots of different manufacturers. They were rejected because of that rancid smell, up to $10 million in lost revenue due to canceled sales. And of course, same thing, reputation problems. They're assumed, customers will now start to assume that the product's going to show up rancid, to the point where these customers were demanding that they figure out why is this product showing up rancid, or we're not going to continue to purchase the product. So pain is immediate, pain is quantitative as well. So we started working with them through our consulting services, and one of the first things that we showed was that there's a unique relationship between lipid oxidation and water activity, where oxidation actually increases at low water activity. So there's a minimum at about 0.3. It corresponds typically with the monolayer, although that can vary with products. But here you'll see that if you go up in water activity, you do see a rate increase for lipid oxidation, and this has to do with mobility, typical reasons why water activity increases reaction rates. At low water activities, as you go down from 0.3 to essentially dry conditions, you also see an increase. And this is quite unusual to this type of reaction. And it has to do with the fact that lipid oxidation requires, of course, oxygen and the formation of free radicals out of on lipids, and that's catalyzed through the presence of metal ions that form these free radicals that catalyze the formation of these free radicals that are then subject to oxidation to produce the rancid smell. At low water activities, hydration spheres, as you remove layers of water to getting to just that monolayer of water, those hydration spheres that are lost lead to an increased access by these lipids to these metal ion catalysts that then result in higher formation of free radicals. And then those free radicals are less likely to be quenched because you're in a condition of such low water activity. And you actually even have higher oxygen diffusion. So all of those combined lead to an increase in oxidation at very low water activity. So when this group decided we'll lower the moisture content to try and prevent this from happening, what they found, so they were in sort of this region. If we look at the isotherm, so this is water activity versus moisture content for their particular baked snack. When they went to lower moisture content, so they tried to push that down to prevent it, they were actually making the whole thing worse. And that's why they saw increased rejections when they went to lower moisture content. When they went to higher moisture content, all of a sudden things started to look better and they were getting good results. And that's because when they went to higher, they actually went closer to that monolayer value of that water activity that was associated with low rates of reactions. And so the product actually remained good. And so without that understanding of how water activity is impacting this, it didn't make any sense. Why would going to higher moisture content result in less problems and less rejections? And so it was critical to have that understanding of water activity and its role. And so really the only way to be able to figure out a safe zone for this product was to look at water activity. We had to figure out what water activity range is safe because oxidation is minimized, but it also has the proper texture and of course would be safe from microbial problems. If they went too low to low water activities, it's going to become unacceptable because of rancidity. If they go to higher water activities, there is an increase in reaction, but the problems would more likely be from texture changes where it becomes too soft or chewy for this particular product. Or if you get high enough, it could be susceptible to microbial contamination. So as we found out, there was a very distinct and specific range where for water activity where this product would be optimal. And so once we had that established, it was clear why their original specification, when they pushed down to low moisture, was not going to work and why they were getting increased rejections. When we adjusted that specification up to a higher water activity, then we can, well, in fact, they pushed it to a higher moisture content, which then resulted in less recalls. By doing our analysis and looking at that range, we can find the recommended spec is actually at a little bit higher water activity, which then translates into higher moisture content so that they have the capability of being able to release this product based on water activity. And as long as it's based on water activity, then the moisture content becomes a standard of identity, even though and prevents there from being a situation where if they were only going off of moisture content, they would have the potential to have problems. So the take-home message is that water activity as a release specification, once it's determined what that ideal water activity range is, it never fails because water activity can be verified through standards to make sure that it's continuing to give good results. So our recommendations in this case was to adjust the specification to 0.5 water activity. This corresponded with 8% moisture, which was actually higher than any moisture spec they had set at that point. So they're able to maximize, again, their return, but also to keep it safe for all the quality and safety factors. So the shift was to then start releasing product based on water activity instead of moisture content. And this is because there's potential errors in moisture content that you don't want to release that product. Instead, moisture becomes a standard of identity. And these recommendations aren't necessarily easily implemented in a lot of these situations where they've been running off of moisture content for so long. You try to introduce water activity as the new release specification, and it can come with some challenges. But in the end, the return that you get for that and the fact that it's critical to being able to eliminate these problems. Again, you might go for years and years with moisture content and never have any problems, but then when that problem does happen, as we've shown here, it's a very painful problem that happens that could be avoided. So critical to both of these situations was the implementation of a water activity meter as a QA test to make sure that these products were in that safe range. Our third example is a nutraceutical powder, and of course we've done some specific examples here with pet food, with a snack, and then with some powders. But these concepts of how to look for product failure and then how to put in safety measures to prevent it from happening would apply to all kinds of different types of products, not just specifically the ones that we mentioned here. And we look at powders; these could be any in a wide variety of different powders. The snacks could be a lot of different dry ingredients or snacks that could be considered. And again, with the pet food, similar to lots of different products that could potentially go moldy. So we're able to use these concepts not just on these specific products, but all types of potentially manufactured products. In this case, we're going to talk about nutraceutical powder that was showing up caked. So they were making this nice powder that was supposed to provide some benefits, health benefits. You basically put it into water and and instantize it to become like a drink, like a protein powder drink, if you will. And when it showed up caked and clumped, due to whatever was happening, of course, it was rejected by the consumers because it's really tough to get those clumps to go into solution. And so you end up with a protein drink that's got all kinds of clumps in it. And those clumps don't necessarily taste all that great. So again, a good reason for customer complaints. So again, see some lost revenue due to these canceled sales because these products were showing up caked. And of course, reputation problems go along with that. Again, you're trying to promote a unique property from these specialized products, but then they're not able to be delivered because of these problems that are occurring after production. So key to this one was understanding why was this product showing up caked and clumped. And caking and clumping, in a lot of cases with these instant and typically amorphous powders, whether that be drink mixes or dry ingredients, when they are in this amorphous state, when they're spray dried or when they're freeze dried, puts them in a what's called a glassy state. And in that glassy state, they are metastable. They can be stable for quite some time, an extended shelf life, and they'll stay free flowing as long as they're down in that glassy state. If we take a look at the isotherm for these products, here's water activity again versus moisture. As water is taken up and the water activity increases, what we see is a very sharp inflection in the isotherm curve. And this indicates when that transition happens, going from glassy to a rubbery state. So this also corresponds with the water activity where this product would start to cake and clump. And so this became a critical part of solving the issue here, was to determine from the isotherm where is that critical water activity. So here we have in these water activity ranges a free-flowing powder, but if humidity is introduced or water is introduced, causing that water activity to increase, and it goes past that critical water activity, that product is now going to be caked and clumped. So the key here is to stop and prevent the water activity of this product from moving above that critical value. And that wasn't, so what wasn't happening in this case was, number one, it wasn't being checked after production to try and get the water activity down in a nice safe range away from where this critical value is, which this critical value will change with temperature. So it will get pushed to lower values as you increase temperature. So both temperature and uptake of moisture would contribute to this. So how do you prevent that from happening to make sure that you maintain below this critical water activity? Well, the main solution here was to make sure that the right packaging was being used on this product. Because what determines once the product's been made and packaged and then sent out for storage and delivery, at that point, what's dictating what's happening with the water activity of this product is all about packaging, plus the storage conditions that it's subjected to. So if it's subjected to relative humidities that are higher than this 0.43, which is 43% relative humidity, and water is able to move into the sample through the packaging, then we'll see that water activity go up past it and you'll get caking and clumping. But if you use a packaging material that serves as a good moisture barrier, and you put the material in there, then now you have something that's going to prevent, even though you may be exposed to high humidity, and you'll see maybe an increase in water activity. The key is that it won't go higher than this critical value. And you can look at what that the water vapor transmission rate of this packaging needs to be in order for this to not happen based upon storage conditions. There's models that can be used to figure that out. We use Fick's first law for diffusion to do calculations based on what is the water vapor transmission rate properties of these packaging materials. And based on that, exposed to a specific condition, what will the water activity change be? How much will it change? We can also reverse this and actually solve for the water vapor transmission rate based on what water storage conditions it's exposed to and what is the critical water activity, what type of packaging, what water vapor transmission rate would you need in order to maintain that product and keep it from going past the critical water activity. And so basically what you're looking at is these permeance values out of the Fick's equation, and you want to find the right permeance value that's going to make sure that this product stays below that critical water activity throughout its life. So the recommendations, first was to determine the critical water activity, which we did through the use of the isotherm. And that set specifications on finished product to make sure that it's processed to lower than that critical water activity, and especially lower at it even under abuse conditions, again, factoring in what the temperature effect is. And then we recommended using packaging modeling to figure out what type of packaging do they need. This prevents both under packaging and over packaging, which both are wasteful, because if you under package, then you have the potential to have problems. But if you over package, you're wasting money on packaging that's more expensive, that when in fact you wouldn't need that high of level of packaging to be able to achieve the results that you want. So those packaging modeling, and we went through the steps of doing that, showed them how to use those models in Excel through the consulting services and set them up so that they could look at those models and figure out exactly what type of packaging they need. Okay, so that's our part one. We're going to now move on to looking at shelf life testing, but do we have any specific questions on those examples before we move on? Let's see. We did have one question come in about what is a packaging model. So the packaging model is this equation right here. So it's this fixed first law. So what we do when we do that modeling is incorporate this equation into, say, an Excel sheet where you can type in all the factors that are known, and then it will solve for what's unknown. Whether that's the water vapor transmission rate or what the final water activity will be, it's just reorganizing the equation so that we can solve for what's unknown. And again, that's all done in Excel, where we can input the things that we know and then make predictions based on that. Okay, any other questions before we move on to our second part? All right, so let's talk a little bit about shelf life testing then. So I showed in these examples situations where these failures happened, and then we had to go and figure out why. And in each case, it ended up that what it was related to and could have been avoided by having a nice water activity specification that identifies the ideal water activity, and then it's being tested routinely as a QA test, and product is released based on meeting that water activity specification. But hopefully, in making a new type of product, those potential problems could be solved and identified prior to actually having the product released, and that's where shelf life testing comes in. And really, shelf life testing should lead to being able to set specifications that are going to make the product optimal. So the way that we address shelf life testing and the proposed paradigm that I'm putting forward is what I call shelf life simplified, which basically takes a water activity based approach at being able to look at shelf life, because ultimately the two main factors that are going to impact your shelf life, external factors, are going to be, well, external and internal, but water activity and temperature are going to be two of the main factors that are going to dictate what the shelf life is going to be. And so if we can, temperature is external, so that can be controlled through temperature control, but the internal water activity, which is impacted by temperature, but also is impacted by formulation, is really where this can be, these shelf life issues can be addressed. So the way that this works is by taking a water activity based approach. The first question is, what is the expected water activity in my product? And so is it going to be an intermediate moisture food? Is it going to be a low moisture food? Where in that range from 0 to 1 do you expect the water activity to be? That doesn't necessarily mean that that's where it's going to be based on formulation or whatever, but as it sits now, what is that water activity expected to be? And that could be found just by making a proof of concept formulation and then measuring its water activity with the water activity meter. And then once that's known, then the question is, at that water activity, what is the most likely mode of failure? And that's dictated by what that water activity range is. So if it's greater than 0.85, as you can see, the main thing that's getting in shelf life is microbial spoilage. These are products that are typically refrigerated. 0.7 to 0.85, microbial spoilage is still a problem with molds, but also chemical instability. Reactions are starting to be at a maximum in this range. And so while microbial spoilage may not be a problem, if the product goes brown immediately or if there's oxidation, and you have off flavors, off odors, then the shelf life of that product is ended. In the 0.4 to 0.7, now we're in chemical instability again, but now we add texture. And these intermediate moisture foods are in a lot of cases multi-component, like a snack cake, where moisture migration will play a role. You get to lower water activities down the 0.2 to 0.4. It's all about texture, caking and clumping, having a snap to it. And then you get to water activities less than 0.2. Really, chemical instability is the problem. So there's lots of products that are made to very low water activity. If they have lipids in them, the thing that's going to end their shelf life is lipid oxidation. It's why potato chips, whose water activity is about 0.1 or lower, have a short shelf life, is because of course, they're cooked in fat and they're very susceptible to oxidation. Okay, so when you're going to go about doing this testing, then based on what we've identified as being that likely mode of failure, the next step is to actually see and track the progress of that mode of failure. How fast is that failure going to happen? How fast is it going to progress to a point where it's no longer acceptable? And so the general experimental design here is to control water activity to 3 water activities and 3 temperatures is ideal. 2 is maybe okay, but 3 is ideal. The way to control water activity, the simplest way is just to use saturated salt slurries, but you can also get nice humidity control packs through companies like Boveda that provide packs that are already at certain water activities. You put those together in a sealed container and let them sit and bring them to a specific water activity. You can set those containers in an oven and set the temperature to control temperature. And then you track the progress of this mode of failure on these samples that are being held at these water activities. And that's you need to come up with some way to quantify that. Sometimes that's sensory related. Sometimes it's an actual chemical test or a laboratory test to figure that out. And then you collect a time series where you've got time versus quantity of change. And when you model that, you're able to come up with a rate constant for each one of those different combinations of water activity and temperature. So how do you quantify chemical reactions? So there are different ways of doing this. Of course, sensory, again, being the most meaningful because it's directly related to what a consumer might experience. However, sensory data is sometimes hard to collect, can be expensive to collect. So sometimes it's better off to use just a chemical test, assuming that chemical test has been related in some way to sensory loss of when things become unacceptable. If we look at lipid oxidation, there's a couple of options here. One of them is the OxyTest. This is actually an instrument provided by NewTek Group that tracks lipid oxidation progress through the consumption of oxygen. There are also other things like peroxide values, T-bar values that can be used. For browning, there are formation of reaction products that can be tracked through chemical analysis, or color changes, looking for the actual browning that's happening. In vitamin loss, you can use test kits, ELISA kits, as well as things like HPLC. Once you've collected this data over time, the next step is to model it. And so we look at kinetic models for these reactions. They can be 0 order, 1st order, 2nd order, although at least portions of each reaction, and a lot of these reactions like Browning reaction have multiple steps, but within those reactions, the step that's leading to whatever it is you're quantifying, in a lot of cases, that step itself will follow either a 0 order or a 1st order. And when they follow these a 0 order, if we plot time versus concentration for that reaction, then and at 0 order, you'll see a straight line. And the slope of that is what provides us with that rate constant, or the rate of the reaction at that given combination of temperature and water activity. And so that if it's a 0 order, we plot concentration versus time. If it's a 1st order, then you'd plot the natural log of the concentration versus time. And again, the slope will give you the rate constant. Then once you have the rate constant values at the combinations of different water activities and temperatures, then you're able to actually put that in and do some empirical modeling to figure out a way to be able to predict what that rate constant is going to be at any given combination of temperature and water activity. So in doing this, it's always best to use data that's collected on your product and not something that's just in the literature on someone else's product, because it's not going to match. And of course, you need to have a way of tracking the mode of failure, whatever is happening. You have to have a way to quantify that and track it. And then once you have those rate constants identified at the different combinations of temperatures and water activities, then we plug that in and create an empirical model that accounts for both temperature and water activity. And that model is called hydrothermal time. So let me show you an example of how that works with baby formula. This baby formula, we took and did an actual investigation, held it at 3 temperatures and 3 water activities levels, and the oxidation itself was tracked using a T-bar method. A T-bar value of 1 greater than 1 indicates unacceptable. That's when it's failed. The reaction that we were tracking followed a pseudo zero order kinetics. And then when we took the reaction rates and we had them at each temperature and water activity combination, we configured those out from our time series. Then we used an equation called hydrothermal time that's derived from fundamental principles, the Eyring equation for rate change, where it tells us what the rate is based on some inputs, one of which is Gibbs free energy equation, or the equation for Gibbs free energy related to water activity. So here's a way that we have both temperature and water activity that we can then incorporate into a rate equation. And by rearranging, we're able to isolate where we can determine what the rate is based on what the rate is at a standard state, as well as then multiplied by several constants. Now, the modeling process we do then is to come up with what these values, these constant values are that fit the equation for a particular situation. So we can do that through least squares modeling and come up with some constant values that are specific to the reaction as well as the medium that it's in, or the matrix that it's in. And so in this case, we came up with values for each of these constants specific to the baby formula, and we were then able to plot the rate, the measured rate by our time series versus what was predicted, and get an R-squared value, which showed very good agreement. So now with this equation in hand, I can plug in any combination of water activity and temperature and calculate what the rate of the mode of failure is going to be under those conditions, which is essentially is our shelf life. So once you know what that rate is, you can calculate how long would it take, given that rate, to move from the level that it's at now to a level that's unacceptable, and that time that takes is your shelf life. What's nice about this approach is that you can also use it to do real-time shelf life. This would be identified as a situation where samples, when they're stored, they don't sit at the same water activity and temperature for the entire time. They're going to change based on packaging, based on conditions that they're exposed to. And when you change conditions, you don't go back to 0 and restart counting again. Because, for example, in this case, we can use the model to figure out that if this product spent 14 days at a 0.5 water activity and 25 C, it will have experienced 25% loss at that point. So it's still got some shelf life left, but it doesn't have all of its shelf life. And then if it spends time at 0.35 water activity for 14 days, it's only 15% loss that occurs. And now we can add these together and figure out what our shelf life is. It's cumulative in that case. And then it spends some time at 5.5 and 25 C, 8 days there. We've now experienced 30% loss because the water activity was high. So in total, we've experienced 70% loss over this time, and our shelf life is the summation of the days spent at each one of these situations. So 36 days is our shelf life up to this point. Now, what our actual shelf life will depend on how long it's going to take to use up this 30% shelf life that's left. And that's going to be dictated by, the conditions. So we can take product at any point and take a measurement and figure out, based on where its value is in our way of being able to quantify the reaction, and figure out exactly how much shelf life is left before it's going to exceed that unacceptable level. And then how long will it take to be able for that to happen, given a specific set of storage conditions. So we can get a cumulative shelf life based on what the history is, or we can take a snapshot and then figure out exactly how many days of shelf life are left for that product based on what value it's at now and what the storage conditions will be. So again, the idea here is to give you some initial information about some of the things that we can provide, but not necessarily everything, because we don't have the time to do that. But we can spend that time with you if you're interested in being able to provide these consulting services, where we can consult with you on how to figure out why a product's failed, or consult with you on how to conduct a shelf life test, or how to create these models, how to model packaging. So we have those capabilities that we can provide that kind of information. And we've done that, as we've shown, for several different customers. And here's just an example of some of the feedback that we received, talking about how we were able to figure out what was going on. We were able to teach them about water activity, and we were able to figure out what was happening, as well as implement specific strategies to be able to prevent this from happening in the future. And so with that, that's my presentation for today. Again, if you are interested in learning more about the consulting services that we offer at New Tech Group, please contact us at the information shown on the page. And I'll turn it back to Orna for any questions we might have.
If anyone has any questions, please write them down in the chat bar. We have a few already, Brady, that came in. Someone's asking if optimal product specifications can help extend shelf life while also preventing waste. I think.
Yeah. So, one of the things we locked in on was the importance of establishing a ideal water activity specification. And then once that's established, then you can prevent over drying, because you dry or you process your product until it reaches that ideal water activity, and then you stop. And any further processing or drying is going to just cause problems. It's not going to be in the ideal water activity range. So there's no reason to use that additional energy, as well as removing your cheapest ingredient, water, from your product. So it certainly is possible to be able to both make your product more stable, but also eliminate any lost revenue due to waste from over processing the product.
Thank you, Brady. Another question: do you use the mode of failure calculations to predict things like microbial spoilage endpoints?
Interestingly enough, there are several different types of models that potentially would be used depending on what it is you're tracking. And for microbial contamination or microbial growth, it is possible to model that through empirical modeling, although the hygrothermal time that I presented is more specific to kinetic processes associated with chemical reactions or other types of modes of failure. There are examples, though, of doing microbial growth modeling where you have several inputs beyond just water activity and temperature, things like whether you have a preservative in place, because there are other factors for microbial growth that affect its ability to grow. Nutrients, we call those hurdles. And so the models that are used for that are going to have more factors included that have to be accounted for. And so in a lot of cases, what you would end up doing there is controlling each of those aspects, and then basically doing a multiple regression empirically derived model to be able to track the rate of growth, and it being specific to the organism as well as specific to the matrix that you're in.
Brady, one last question really briefly, because we're about to end. If you can describe how the water activity meter works, the IR measurement.
So the water activity meter itself, very simple. Water activity is the equilibrium relative humidity that's above a sample. So to measure water activity, you put the sample in the instrument, you close the lid, you push the button, and it's basically reading the relative humidity inside that chamber, waiting for equilibrium. And so at equilibrium, the vapor pressure in the headspace is equal to the vapor pressure of water in the sample. And so at that point, if you measure the relative humidity inside that chamber, that equilibrium relative humidity is the same as water activity. And that's how all water activities work. It's just they only differ in how they measure what is that equilibrium relative humidity. Some measure it through an electrolytic sensor, that like the instruments from NewTek. Others use other types of sensors to still measure that same equilibrium relative humidity. And that process, the length that it takes is very dependent on the product, of how long it takes for it to reach equilibrium. And that's really what determines the time of the test. The testing process itself is similar for all of them.
I see there are a few more questions, but we have to wrap up. So we're going to get back to you offline. And we encourage, everyone on here, if you're interested in sharing what you do and what may be some of the challenges that you're facing as they relate to shelf life, please reach out to us through the information that's on the screen or through our website. Before you leave, Eden's going to post a survey. The survey is posted already in the chat forum. We'd appreciate if you could share your input about the webinar today. And we hope to see you in future webinars. Thank you again, Brady. And thank you all for joining us today. Enjoy the rest of the afternoon.